So here's what I'm going to do is, when you guys get done, when you've correlated your genes and you've found your top 1,000 correlated, be sure to send that list off to everybody. So if you look here, so I've been keeping mine in my own folder, big data, lung cancer data sets. You can call yours whatever you want. But if you look down here, these are the analyses that I put in here. Let me see. So here's my list, Dr. E, I've got BTs, MOs, and condors. Okay? Because eventually what we're going to do is we're going to download everybody else's and then basically combine them and then, again, figure out which genes were around all these different banks that were robbed.
How do you add those in there like that?
What's that?
Is that like the actual file?
Yep, that's the actual file. So you just import, just like you did with your gene list.
So it's the same thing we did as with ours, but we're doing it with like
Yes.
BTs or MOs.
Right, and remember you have to know which platform they used. Remember you have to click the Affymetrix or Illumina, depending on which platform you use. That's exactly what we're doing. So here I'm going to pull up mine just to kind of go through, here's what I do. And I'm going to show you how to do a comparison analysis. So be sure after today I want you to download the gene lists that were already put out. So MO, BT, and Condor all put their gene lists out.
You sent to the Georgia, not to your...
I did today. Should have got that today. Okay, so here is basically the summary page. Okay, this will be the first tab that you'll see. This just kind of gives you all the kind of top 10 of all the different tabs that you have. And I'm not going to go through this because we're going to go through the other tabs. So staying with that presentation I just gave you, let's look at criminal histories. So I go to disease and function tab. Okay, this looks familiar, right? Given here, so we can color this any way we want. By z-score, now you're coloring it by its activation state, or at least what the software thinks. We can also color this by the significance of the findings. So you can see this stuff here is very significant. You wouldn't expect this by random chance. I like the absent present or the activation, so I'm going to go z-scores. Now here's what you can do is you can size by, I'm going to size by the number of genes. So the bigger the box, the more genes from my top 1,000 it contains. So if I go over this box here, this cancer, there were 208 genes associated with cancer out of 1,000. Okay? And if I keep clicking on that box, I can get up to any of those genes. Let me click it one more time. Show you what you'll see. So again, this is criminal history, is what this kind of page is. Who's got the criminal history? Who's been guilty of cancer before? Just because they've been associated with another cancer doesn't mean it's playing a role, but increases the odds, doesn't it? So given here, and you can download any of these lists. Oh no, there it is. So this is what you'll see. These are all the genes according to the software, which is based off of the entirety of the scientific knowledge that has been published. Basically shows that this gene, when it goes down, tends to indicate an increase in cancer, right? And here's the findings here too. You can go to any of these sheets or any of these references and look up the actual papers where it says this relationship. So again, there's a lot of genes here. It doesn't mean they're all guilty, but the likelihood that they are kind of goes up. Okay? And here's the log ratio here, and then the findings. Okay, let's go to the top again. Here's what's cool. Remember that thing I showed you, the invasive bladder tumors? It looks kind of the same way, right? Got a lot of cancer that's up, a lot of, again, cancer. Cell death going down. That's the same as bladder cancer. And when I look at these cancers, I see similar patterns that I think a lot of people mischaracterize. Like if you say lung cancer and bladder cancer, I find some types of bladder cancer more like lung cancer than they are to other types of bladder cancer. And it's and most people, they just divide these cancers, but I think these cancers are very similar. Again, cell movement, cell growth. This looks a lot like what we looked at with the bladder cancer. And again, you can go here and look at any of these relationships. Okay, criminal history. Now let's look at our gangs. That's canonical pathways. Anybody know what canonical means?
Biological.
What's that?
Biological.
By and large?
Biological.
Biological? No. Anybody else?
Carnivorous
No.
creatures.
Canonical means like established, like this is what a bunch of experts said. This is the idealized pathway. And like I said, these gangs are just as good, they're only as good as, the information we have on them. So here's what you can do with these pathways. I like to display it horizontally because you can read it, which is always good. Anything with an orange would tell you that pathway is probably up. Anything blue would tell you that pathway is down. Oh, this is so awesome. So let's look at this first pathway. You can click on this bar here. So if I click on it, now if you look down here, all the genes associated with that particular pathway are given below. Okay? Now we can look at the pathway. Let's look at it. Maybe. Chunka, 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 chunka. Remember, you're just sending instructions to a computer in Silicon Valley. So that's why it's delayed. All this is being done in California.
Yeah, this looks colorful.
Yeah. This makes sense, right? They're showing this is more active. Look at all these down themes or things that are up-regulated. But here's the cool thing, is if you look here, these two signaling molecules are down. And these get signaling from the outside of the cell. So even though this is all up, it seems like the signaling molecules are all down. If it has a double line on it, that means it's a complex. So if I click here and if I right click and I say show members, this will show me all the row things that were in my gene list. So anything colored was in your gene list. Anything that is not is not. And if we look here, you can see these 3 row things are up. And this one is down. Row T1. That's unusual. These are things that you look at. So here's what I could do is I could say, okay, RHOT1, I'm going to look that up. RHOT1. And again, you can just Google this stuff. This is how easy this is. Here's what I like. I like to look at NCBI. That's usually a good place to look, the gene list. So we can click on this. This is human. This is what RHOT1 stands for. Come on. And this just gives like kind of a, ooh, what happened? Wait a minute. I'm kind of missing part of my window, so it's kind of hard to, there it is. So here is a gene, and if you look down here, this will give you all, this is like its aliases. This is basically its rap sheet. This is the crime database that you have on this guy. If we look down here, so this gives you some of the recent articles. This is kind of what it does. Gene references into function. If you look here, there's a lot of cancer stuff. Where's the one that I saw up here? So row T1 and SMAD4 are correlated with lymph node metastasis and overall survival in pancreatic cancer. If I click on this, and if we read this, basically what it's going to tell me is that when this gene goes down, the cancer gets much worse. And this is in pancreatic cancer. We're looking at lung cancer. We see the same trend. That's exactly what you're looking for, right? This is important. Not only did it rob a bank, or was around a bank that was robbed, it's probably one of the good guys. It was probably trying to help, right? This is probably a tumor suppressor that we've just now identified with lung cancer. And I doubt anybody else out there knows this. We just figured out something that no one else in the world knows just by messing around with a bunch of numbers. That's what you're looking for when you analyze these things. Does it make sense? And if you look down here at some of the things that are going on, actin filament stabilization, formation of stress fibers, this is all actin polymerization. This is all cells breaking down their cytoplasm to keep dividing. This is what this basically shows. Not only that, but here's the information that I get is this signaling to break down the cytoskeleton to divide more isn't coming from outside the cell probably. It's probably internal. That's one thing to keep in mind when you look at these is remember we're looking at cancer. If everything was going exactly like it was supposed to, then there wouldn't be cancer, right? That's kind of what you're doing is when you're looking through this is how does this lay out and what doesn't make sense? And it's the things that don't make sense are probably the most interesting to really drill down into. All right, let's look at, okay, then there's some other cool things in here. Cell adhesion, you're going to find a bunch of cell adhesion stuff. I love this. Okay, come on. Look at all this stuff going up. I mean, this makes sense. And some of this stuff is going down. It's so weird. Like, this pattern will be different when we look at formation of lung tumors, but you know, this is what's going on. These are the changes in the cell membrane that this cancer needs to undergo in order to get worse. Let's look at networks. So these are the gangs. They can give you a pretty good idea of what's going on, give you a general flavor, but they probably don't spell out the specific relationships. So let's go to networks.